Well, good afternoon and welcome to this first of our UCLH BRC COVID research seminars. I'm Brian Williams and I'm going to chair this session, and in a moment I'll be introducing the panel and our speaker for today. Just a few housekeeping notes before we start. The seminar is being filmed, and the reason for that is we want to make it available on the BRC website for those who've not had the opportunity to view it if they wish to. The audience will be deliberately muted throughout, and you can't actually turn your cameras on, so don't worry about that. However, you can ask questions, and there is a Q&A function that you should be able to see at the bottom of your screen. And make sure when you click on this that you use the section which says questions to panelists and the audience. So because we'd like the questions to be seen by both the panelists and the audience. And then we will be skimming through those and picking them up as when we get to our Q&A session, and we'll try and get through as many of these questions as possible. And with that in mind, don't wait to the end before submitting your question, as we'll get deluged suddenly. If there's things that you hear as you're going through and you think about you'd like to have some clarity on, please put a question in that question box. So I'd like to introduce the panel. I'm sure they're well known to all of you. David Brealey, who is a consultant in intensive care, obviously very busy, and good to have you here, David. And so he's representing a sort of intensive care point of view on the panel. Chris Hasford, who many of you will know, works in acute medicine and also respiratory disease, so we have respiratory representation. And Maddy Noursadeghi, who you'll know, is also doing work in infectious diseases and acute medicine, so looking at this from an infection perspective. But most importantly, today we have one presentation, and that's going to be given by Rishi Gupta, and he's one of our star academic trainees in respiratory medicine and is an NIHR funded clinical research fellow based at both the UCL Institute for Global Health and the Division of Infection and Immunity in the hospital. And he's really developed expertise in predictive modeling using routinely collected clinical and epidemiological and laboratory data. And actually, before COVID happened, I think we'll be talking about before and after COVID for many years. Before COVID, his focus has been on trying to predict risk of active tuberculosis, leading to the development of a personalized risk calculator for patients with this disease called Periscope TB. And that was so impressive, that work, that it was published in Nature Medicine earlier this year. And with the onset of COVID, Rishi turned his attention to COVID. And I recall a number of conversations in the early days when he was looking specifically at the UCLH EPIC electronic data and looking at various predictors of outcome. And at that time demonstrated that age and oxygen saturations were really better than any of the early complex models to predict outcome. But since then, and with the availability of the rich data source from ISARIC, he's gone on to use the UK-wide ISARIC database in thousands of patients to develop a state-of-the-art risk prediction model to predict deterioration in hospitalised patients with COVID. And this also is really high impact, published recently in Lancet Respiratory Medicine. So I'm going to hand over to Rishi now and ask him if he'd load up his slides, and look forward to hearing his presentation. Rishi.
Thank you very much, Brian. Can I just check you can see that all right?
Yes, we can.
Brilliant. Okay. Oh, thanks very much for the introduction and for the invitation to speak today. I thought it would be helpful if we start with some brief background to set the scene, and that's really to revisit the question of why we need a prognostic model for people with COVID-19 in the first place. And I think there are a few potential applications that warrant a bit of discussion and may sort of inform further discussion after the end of the talk. So firstly, we might think that a prognostic tool could help us to target our medical therapies towards people at higher risk. And this might be particularly useful for drugs that are subject to resource constraints, such as remdesivir, along with those that have significant toxicity profiles, but also, and perhaps in particular, where we know that the treatment effectiveness of any therapy is modified according to disease severity. Second, better prognostication might help assist our allocation of resources in terms of hospital, HDU and ITU beds. And third, it might also be a mechanism to stratify eligibility for interventional trials for drugs with ongoing equipoise. And we've also heard through informal local feedback from colleagues that the 4C mortality score, which I'll summarise a bit later, has already proven to be quite useful in guiding discussions of prognosis with families and patients early during admission. And so this is an extremely busy field. This is a living systematic review led by a team in Utrecht that's been published in the BMJ and was due to have regular updates. And even by June last year, there were already 50 candidate prognostic models reported in the literature. However, almost all of these were thought to be at high risk of bias due to being derived from small and single center studies and due to often inadequate methods used during model development. And so as Bryan mentioned, we went on to test the performance of 22 of these 50 candidates in this paper published in the European Respiratory Journal. This was using first wave cases from UCLH, where we found that none of the multivariable models in our analysis performed any better than simple univariable predictors, specifically age to predict mortality and admission oxygen saturation to predict in-hospital deterioration. And so we've been working on addressing this issue in collaboration with the ISARIC study team. And many of you will be familiar with the 4C mortality score that was published in the BMJ a few months ago. And the variables that contribute to 4C mortality are shown on the right here. Unsurprisingly, there is a very strong association with age, which makes up a third of the total points in the score. And so we thought that the resource of the ISARIC 4C data would provide a great opportunity to extend this previous work further through development and validation of what we've called the 4C deterioration model. And the rationale for this is that whilst we know that age is a very strong predictor of mortality, we imagine that estimating deterioration risk in parallel to mortality risk may also be useful in clinical practice, particularly among younger patients where there will always be somewhat of a glass ceiling on the mortality risk. We also thought that this would be a nice opportunity to provide a probability output model to run in parallel to the point score approach for 4C mortality. And we were really keen to exploit the wide geographic coverage of ISARIC 4C by exploring potential geographic heterogeneity across the NHS. And I'll explain that further later on in my talk. So the specific objectives for this analysis were to firstly try to develop and validate a prognostic model to estimate risk of clinical deterioration among adults hospitalised with COVID. And then we then sought to benchmark this new model by testing the hypothesis that the new model leads to greater clinical utility than pre-existing prognostic scores. And so, as I mentioned, one of the things we were really keen to focus on in this analysis was to make the most of the wide geographic coverage of the ISARIC 4C study, which is collecting clinical data on patients hospitalised with COVID across the UK and from over 250 hospitals. And to give you an idea of scale, there are now well over 100,000 participants recruited to the sort of clinical data arm. And so we specified a priori to analyse the data stratified by NHS region according to standard NHS classifications. We included 8 regions in our development cohort as listed here, and we held out data from London for further additional validation. So London data were not included in the model development cohort at all. I'll briefly just run through our key methods. So our population here is consecutive adults enrolled to ISARIC 4C during the first wave. So this is between a window of February and August, but many of the admissions, as you can imagine, were focused around March, April time. Our composite outcome here of clinical deterioration encompassed either initiation of ventilatory support through the mechanisms shown, HDU or ITU admission, or death, and this roughly aligns with the WHO clinical progression scale score of 6 or more. We used a logistic regression modelling approach and used restricted cubic splines to account for non-linearities. And we started with a number of a priori candidate predictors, as for the 4C mortality model, and then performed some backward elimination to try to only retain a relatively parsimonious list of the most valuable predictors. And we sort of tried to adhere to best practice throughout in terms of methods and reporting standards. So we used multiple imputation in our primary analysis to account for missing data and adhered to the tripod prediction modeling reporting standards. And this slide summarizes our candidate predictors of interest, which we defined a priori. This was based on predictors that were considered to be associated with differential risk, either among people with COVID or other respiratory infections from the wider literature. And we also specified here, in the interests of sort of pragmatism, that the predictors had to be available in at least 60% of participants in the data set for further analysis. And that allowed us to therefore impute the remaining missingness fairly robustly. I'll briefly walk you through our analysis pipeline. So we started with our 4C clinical data and divided it geographically as I outlined. We then identified our candidate predictors of interest and performed multiple imputation, and all of our downstream analyses were done in each imputed data set and then pooled. Next, we did our backward variable selection, so essentially removing variables that were not adding any significant discriminatory values to the model, and trained the final model using the remaining selected predictors. And finally, we did two types of validation here. Firstly, what we call internal-external cross-validation within the P8 development cohort regions. I'll explain that a bit later. And then secondly, we did held-out validation, as I mentioned, just in the London cohort. So I thought it would be helpful to just briefly summarize the validation metrics that we're interested in here, which is firstly to assess discrimination of our final model, which we quantify as the C statistic. And this is equivalent to the area under the receiver operating characteristic curve, which many of you might be familiar with. So just for illustration, a perfect test is shown here in green with an area under the curve of 1, while a coin toss is shown in red with an area under the curve of 0.5. We're also interested in model calibration, and this assesses essentially agreement between our predicted and observed risk. We assess this through visualizing calibration plots, as shown here with a line of perfect calibration shown in orange. And we also quantify some calibration metrics. So the calibration in the large, which reflects the intersect here, and the ideal metric there should be 0, and the calibration slope, and we aim there for a slope of 1. But we also sought to go beyond these discrimination and calibration metrics to assess the potential clinical utility in practice. So to do this, we use something called decision curve analysis, which I'll spend a bit of time explaining here, as I think it's quite a useful concept, if somewhat abstract, to get our heads around. So in decision curve analysis, we calculate a metric called net benefit, and we plot this visually. So this is an example which is taken from a previous article in the BMJ a few years ago. And here decision curves are shown for a range of strategies in the different colors. So recommending an intervention for everyone in blue, regardless of any test results or risk prediction models, using a risk prediction model in red, using a biomarker in green, and then recommending the intervention for nobody in brown. And the intervention here could be any number of things, such as an escalation of treatment with a specific medicine, for example, or in other cases, it might correspond to further confirmatory testing if you're doing some initial screening. And we calculate net benefit here on the y-axis as the trade-off between correctly identifying true positives and incorrectly identifying false positives. And these false positives are weighted by something we call the risk-benefit ratio, which I'll explain. And so the risk-benefit ratio here is represented across a range on the x-axis as something called the threshold probability. And this essentially represents the cutoff for our risk prediction model output, above which you might recommend any given intervention, such as medical therapy. So it's helpful to show this by relabeling our x-axis. So a threshold probability of 1% corresponds to a risk-benefit ratio of 1 to 99, meaning here we think our benefit of our intervention is likely to be 99 times greater than the risks, while a threshold of 5% would indicate a risk-benefit ratio of 1 to 4. We can also relabel the x-axis as the number willing to test or treat for each true positive case that we identify, depending on what our intervention is. So a threshold probability here of 1% corresponds to a number willing to test or treat of 100, and a threshold of 5% means a number willing to test or treat of 20. And finally, it can also be useful to relabel the x-axis according to the clinician or patient preference. For example, for people who are very worried about the disease and are less concerned about the intervention, the threshold probability is likely to be quite low, while it would tend to be higher if the clinician or patients are more concerned about the adverse effects of the intervention. So just some brief examples. The vertical dashed line in grey here indicates the threshold of 1%, so a number willing to test or treat of 100. The strategy with highest net benefit here is indicated in the blue line, so that means intervening for everybody regardless of their biomarker or risk prediction results. In contrast, the second grey line indicates a threshold probability of 20%, so a number willing to test or treat of 5. And here, the strategy with the highest net benefit is to use the risk prediction model. But broadly, in this decision curve analysis, we are really looking for the approach that leads to the highest net benefit across a range of clinically plausible thresholds for any given decision. So broadly here, using the risk prediction model looks like it's the best approach across a range of threshold probabilities. So on to our key results. I'll firstly just briefly summarize our baseline demographics, and they're shown here stratified by the first chronological way in which participants met our composite outcome. So we included almost 75,000 participants in this analysis, of whom around 43% met our composite outcome. And as you might expect, participants who met the outcome through the initiation of ventilatory support or HD or ITU admission tended to be younger than those who met the outcome because of inpatient mortality. This histogram gives us an idea of the timing of the outcome events as days from recruitment to deterioration. As you can see, the deterioration risk declines markedly with increasing time since admission, which isn't surprising. And many of these early deterioration events represent initiations of ventilatory support or HD or ITU admission. Now, these plots show the 11 predictors that were included in our final 4C deterioration model, along with their associations with our outcome. So these are all things that we routinely measure at the point of hospital admission, and we are reassured that they all have plausible associations here with our outcome of deterioration. And we've standardized the y-axis here, which is the risk of deterioration, so that we can get a feel for the relative weightings of each of these 11 predictors. So unsurprisingly, we found that the oxygen saturation on arrival is the strongest single predictor of the 11 included in the final model. Now, this schematic summarizes the internal-external cross-validation method that I mentioned briefly earlier, with each rectangle here, in this example, representing an NHS region. So this shows that in each iteration of this internal-external cross-validation exercise, and the iterations are numbered on the far left, one of the regions is iteratively removed from the development set and instead used for validation. So this gives us 5 sets of validation parameters, which we can then pool to come up with pooled estimates for our discrimination and our calibration metrics through random effect meta-analysis. And so we think this is a really nice way to try to get a feel for the between-region heterogeneity and thus the potential generalizability of our final model. So here are the 3 forest plots for each of our 8 NHS regions within the development cohort. You'll see that the C statistics are very consistent across regions with a pooled estimate of 0.76, indicating very consistent discrimination. The calibration slopes on the far right are also very consistent, with pooled estimate of 0.99. The calibration in the large in the central forest plot is a little more heterogeneous, and there's some minor variation here due to varying baseline risk in some regions. For example, there was some underestimation of risk overall in Wales and South East England, and some overestimation of risk in South West England and Scotland. But overall, the degree of miscalibration here appeared quite minor. So we were also reassured by that. So we then took our final model, trained across our 8 development regions, and validated it using our 11 predictors in held out data from over 8,000 participants in London. And here I've benchmarked performance of the final model to other candidate prognostic scores. So you can see here that the 4C deterioration model had the highest C statistic in our validation cohort. and again, with very consistent validation metrics in terms of calibration, as we saw from our internal external cross-validation. On the left here is the London validation calibration plot, which looks nice as it's very close to the line of identity. And on the right is our decision curve analysis that I described earlier. So here the key take homes that you can see that the 4C deterioration model in solid red had higher net benefits than the other candidates' scores, and also had higher net benefit than either the treat all and treat none approaches across a broad range of threshold probabilities. And this to us provides really encouraging evidence of the clinical utility of the final model for clinical practice. So finally here, in order to put the new deterioration model in the context of our pre-existing 4C mortality score, we've done some comparisons of their predictions in our London validation cohort. So here we've plotted the predicted deterioration risk on the x-axis versus the predicted mortality risk on the y, the mortality risk being from the 4C mortality scores, and the colors here indicate age. So the main take homes here are firstly that the deterioration estimates tended to be appropriately higher than the mortality predictions overall, but these differences were magnified among some of the younger patients in the cohort. So this sort of bottom right part of the scatter plot here, who can often be at quite low risk of mortality, but despite quite high risk of in-hospital deterioration. And we think it's a really important point to emphasize why it's important to consider parallel models for each outcome together. And to further illustrate this, we've provided 10 example patients here. The most discordant patient is patient number 8, who has a low mortality risk of around 10%, despite a high deterioration risk of around 60%. And if we look at their characteristics in the heat map below, we can see that they're fairly young, so age 56, but with clear evidence of acute illness with a high respiratory rate, requirement of supplemental oxygen on arrival, and a high CRP. So we've now implemented the 4C deterioration model alongside the 4C mortality score on the ISARIC 4C website. The user needs to enter the inputs required for both models, so it's 12 inputs in total, and the site will then generate a nice infographic as shown on the right here that presents both the mortality and the deterioration risks side by side. And these are put in context in the infographic of the distributions of risk in the wider ISARIC validation cohort. So I'd just like to briefly summarize some summary points before I talk about some next steps to inform the further discussion. So here we've developed our 4C deterioration model, which integrates 11 routinely measured variables, each of which has clinically plausible associations with the outcome. We found that the model demonstrates consistent discrimination, calibration, and clinical utility across all 9 NHS regions in England, Scotland, and Wales, so it indicates strong potential for generalizability. And we feel that this complements the 4C mortality score nicely and may be of particular added value among younger patients for the reasons that I outlined. We do, of course, have some limitations here. Firstly, we are obviously keen to pursue external validation beyond ISARIC 4C and potentially in other countries. Secondly, we're aware that baseline risk is quite likely to change over time, so we may well need to recalibrate our model, and we have a plan in place to do that prospectively should it be required. Thirdly, there was too much missingness for some potential predictors of interest, such as LDH and D-dimer, which there's been a lot of interest in, so we're unable to address those predictors in the current analysis. And we also weren't able to integrate cytokine, proteomic, or transcriptomic measurements in this analysis, which kind of makes sense because we're focusing on a pragmatic and implementable clinical prediction model, but we're certainly planning on extending this work in the future to include the cytokine and molecular measurements for the patients within ISARIC who have blood sampling as part of the tier 1 and 2 parts of the study. And so the paper is now published in the Lancet Respiratory Medicine if anyone would like any further details. Before I end, I thought it would be helpful to touch on some next steps to inform our further discussion. We are, of course, interested in looking at how the models are performing in second wave data using ISARIC 4C in the first instance, and this analysis is underway. We're also working on molecular biomarkers such as blood RNA signatures for deterioration to see if they may add incremental value over and above the routinely measured predictors. And we're really interested in extending our assessments of the potential clinical applications and clinical utility. One theme here that we are particularly interested in is to consider perhaps a post hoc analysis of randomized controlled trial data to see if there's any evidence of effect modification to suggest that the 4C scores may be good ways to stratify treatment with drugs such as steroids, remdesivir, and anti-IL-6, since we already know that the effectiveness of these therapies is likely to vary according to the severity of disease. And I'd be really keen to hear any feedback on this idea, in particular during the discussion. And so I'll end there with my acknowledgements to the huge number of people who've contributed to the data collection and analysis, by thanking you all for listening, and I'll look forward to further discussion.
Well, Rishi, thank you. That was terrific, and actually really complex for people who aren't familiar with this kind of approach to risk prediction. And I think you explained it really, really well. Okay, so there's a few questions come through already, but maybe before I get to those, I just wondered whether there were any initial comments from any of our panelists. As I can see, you can just raise your hand. Any comments anybody would like to make? David.
Hi. Rishi, amazing work, particularly under such pressure and short amounts of time. That's incredible. Now, I'm a bit of a fraud here because my understanding of risk predictions is shocking. But I've been around databases and data scientists for a little bit too long now. And the one thing that really worries me about this sort of work is the quality of your data. You've got a lot of it, but how can you be sure that your data quality is up to standard? I'm just sort of a couple of examples, just looking through your demographics, as it were. Nearly 70% of patients coming in were on air. It's not generally what we see at UCH. Now, maybe it's different somewhere else, of course, and I appreciate that. The respiratory rate, somewhere they're all even numbers, which is absolutely typical for nurses noting things down. And nurses tend to find a median around 18 to 24, which your numbers are a bit higher. But again, I do worry that some of the observations that have been taken down under pressure and at speed are perhaps not as accurate as the reality. And just the final bit on that is, it says the first day of hospital admission, but what does that mean? Is it the first moment they walk through the door? Is it the worst measurement? I assume not. Do we have any idea where this data is coming from? I'll be quiet at this point.
Great. So Rishi.
Yeah, thank you. I think they're really important questions to consider, and something we've been really mindful of throughout the work too. I think there's been a lot of general work done towards cleaning up the ISARIC 4C data set. There's been a huge amount of work done by a core team in Edinburgh, who've done lots of initial QC and some local validation too. I think I personally have a reasonable degree of confidence in the final data. I think we've done some initial crude looks in terms of consistency of the findings, broadly speaking, of the ISARIC 4C overall data set with our locally captured data from UCLH. And there are some really consistent messages there, which is really reassuring. Secondly, I think, as I mentioned when I showed you the associations of each of our final exposures in our model with the outcomes, I think they're all entirely clinically plausible. And I think that's a really important sense check to sort of to verify our findings along the way. So I think that was very helpful to see too. And in terms of your specific points about when these predictors should be measured, again, that's a really important point to stress, that the way they should have been being captured in the ISARIC 4C case record form is that they are essentially the parameters measured at the point of arrival, or as close to arrival as possible. And that's how we would recommend implementation in practice too. So the idea is that you can quickly get a sense for this patient's in hospital risk using their admission parameters.
David, I would argue as well that I agree with your point, that larger data sets don't necessarily compensate for poor data. But actually, in many ways, this has more validity in the sense that the data that is being used to create the score is the data that's being collected on which we base our clinical practice and our clinical decision making, accepting that it's not always perfect. But it is the data that we have in front of us in EPIC when we look and see what these values were when people arrived in the hospital. Chris, you've got your hand up, and then Maddy.
Yeah, thank you. Can you just make sure, can you hear me?
Yes.
Yes, super. First of all, Rishi, I like very much to echo what David said, in the sense that I was hugely impressed when I read the paper that you managed to get 75,000 people, which obviously hugely helps. And just echoing what you just said, Brian, it will automatically sort of help if data acquisition isn't perfect with these clinical parameters we normally do. I just wondered, because obviously it's a real concern, data points. It's a real concern if it comes to oxygenation. I had similar wonders. I also had similar wonders with temperatures. I was very much surprised because in my experience, people who came through the front door had very high temperatures very frequently, yet here the temperature range is between 36 and 38.1. So in consequence, I wondered whether for the second half of the pandemic, you are aiming to achieve what I hope we achieved at University College, whereby we have sort of agreed that we get a set amount of data, for example, blood testing, that we get on everybody a D-dimer when they come through the door. Whether you have now a sort of system whereby all participants use the same agreed lab tests as well as data points, i.e. that oxygenation is measured with everybody by the time they come in, temperature is measured before they get on paracetamol, and so on and so forth. Thank you.
Really good point. Maddy.
Yeah, just to follow up, I think both Dave's and Chris's points, clearly collecting data at very large scale in order to generate generalizable models was the sort of purpose or the order of the day at the beginning. But going forward, just to pick up on Rishi's points about next steps, it's going to be really important for prospective discrete validation exercises in specific contexts, where in fact we have much more detailed oversight of the quality of the data. And I think that particularly EPIC gives us a great opportunity to test this in real life, with anger, to see whether if we can nail these numbers and data more accurately, whether the model still stands up. And I really encourage to sort of, as Chris described, embedding it in our routine practice so that we can evaluate how well it performs.
Yeah, and I also like the fact that you had a focus not just on mortality, actually, because actually whether somebody's going to die or not, we do our best to try and influence that. But actually, what is very important clinically, and particularly the speed at which some of these patients can deteriorate, is trying to predict who is likely to deteriorate, because that might determine where we put that patient and also the level of observations that we might give that patient. Because we've all seen patients amongst the group here all look after these patients, where you're often surprised that in an hour or two after seeing somebody, there's been a sudden deterioration. And if we could pick that up, any characteristics that might pick that up, that would be really helpful. Rishi, we've all commented. Anything you'd like to say in return to those sort of collective comments?
No, I think you've summarised the key issues really nice. I suppose the only comment to say is that this is, on the whole, quite a pragmatic national level study. And the reason ISARIC 4C has been so successful with their tier 0 recruitment is because of their pragmatism. So I totally agree. It'd be really helpful if the admission parameters and the admission blood test could be standardized as much as possible. But actually, ISARIC 4C is recruited for over 250 hospitals. So actually doing that in practice is easier said than done. So therefore, I'd echo Maddy's point that at least if we can get those things right on a local level, then that's a really useful starting point to get a better handle on the further prospective clinical utility of these tools.
And actually, interestingly, and I've done some work in this area, not as sophisticated as you've done here, when we develop news. And one of the things that news did inadvertently was actually increase the focus in hospitals on the need to collect data properly on basic physiology. So, what used to happen in hospitals is, people used to go around making these measurements, writing them on a chart, and few people looked at them, or indeed they were not collated to try and give an estimate of what was happening to that patient. But now, because people are looking at the values and the aggregated values in score systems, there's a much greater focus and a greater emphasis in training, actually, of the people who make these measurements routinely in getting these measurements right. I mean, I think we'll always struggle to get respiratory rate measured properly, particularly as we can't wear watches anymore. But it is a challenge because, if you look at the data physiologically, and that's what I'm getting to my question now, Rishi. The data physiologically is respiratory rate always comes out in all of these risk markers, whether you're talking about trauma, whether you're talking about sepsis, whether whatever you're talking about is the insult that's potentially life-threatening. Respiratory rate is a really powerful signal, and it's probably the thing we measured worst of all. I mean, what was the strength of respiratory rate in your analysis?
Yeah, so we did actually see a nice and clinically plausible association between respiratory rate and the outcome. So it was a really important predictor to include in the final model. But I entirely agree with all the points made so far. There's a lot of noise in that data, but perhaps we've been fortunate here because of the scale of the data set. We've still been able to see a nice and very plausible signal with the association with the outcome.
So we've got a few questions coming in now, and the first one actually is a really interesting question, and it comes from Dave Ramlakan, who's not a medic, works on a lot of our data in the background, and it's a great question, Dave. Do you need different prognostic models according to the COVID variant? But maybe spinning that question around slightly another way, is there any evidence that the sort of risk of deterioration and the mortality risk in the variant of concern that people have talked about, have we got any data on that, about this model's performing? Have you actually got that information on the ISARIC patients about which variant they've got? Are we linking the genomic data with the basic data? Rishi, you're on mute. Rishi, you're on mute.
Sorry, I saw Maddy just unmuted. I thought I'd let him go first.
So I guess I might say that in the ISARIC 4C data set, we haven't reached the point in time temporally when the data for the variant of concern is coming through yet. So as Rishi showed, we're probably at about the end of November at the moment in terms of data completeness to be able to make that assessment. But those data are coming through, and the link is being made with COG UK to be able to make that assessment. So just from the impression I have from existing sort of contemporary reports of the pattern of illness, I guess my guess is I don't think it'll have a major difference in terms of severity of illness, but I think it's too early to say.
And it's also quite difficult because, of course, the first wave was the first wave when we didn't really have many treatments, whereas the second wave is to some extent being influenced by both better clinical organization and practice, but also enhanced therapeutics. So there will inevitably be a difference in outcome in general, but it'll be useful if we could tease that out. The next question, and I think it's also a very important question from Pooja Mehta, is really, and first of all, he comments on the fact that your work is brilliant. But secondly, the really important question that quite a lot of patients, and actually this is probably very relevant to UCLH, are either immunosuppressed because of their disease or are on immunosuppressive drugs. Is it possible to tease out whether, in many ways, being on an immunosuppressant, which might suppress, for example, CRP, may alter some of the other parameters you're measuring, does that screw up the test? I mean, the score, or have you been able to look at that and see whether there's any clinical features, background comorbidities that in any way influence the score?
Yeah, thank you. That's another really important question. To be honest, we haven't been able to look at that with the ISARIC 4C data yet. As you can imagine, in that kind of pragmatic data collection exercise, the sort of medication history bits tend to be quite messy to get through. So I think there have been some quite big efforts to clean up those data fields over time. So it's potentially something we might be able to look at in the future. Alternatively, I wonder whether actually testing that on a more local level with more high resolution data sets that capture sort of drug histories more accurately might be a better way to go there. For example, looking at stratified performance among that subgroup specifically. I think it's a really important question. I don't have an answer for you yet, but there are ways that we could go about trying to address that in the future.
Okay, and then the next question is from Jeremy Brown for infectious diseases. So Jeremy's asked the question that obviously the score is used on admission. Is there any difference in the predictive accuracy between, for example, early scoring and later scoring, or indeed the temporal, day-to-day scoring? Is there a way of looking at that, whether, for example, the trend in a score might influence severity of risk rather than just the absolute value? Is that something you're planning to look at, or is it too complex?
Yeah, really important question again. And I think actually that would be a really interesting sort of extension to look at whether you could use these scores, akin to how we use NEWS2 scores in practice as a dynamic measure of risk. So we haven't looked at that. And again, I'm not sure that the ISARIC 4C data would be the best way to do it because, for example, the daily case record forms are often only captured, day 1, 3, 6, and 9 sort of thing. So I think the way to address that question more definitively would be to use a high resolution electronic health record data set. And there would be the ideal opportunity to do that using EPIC at UCLH, for example. So it's another priority for future work that would be really interesting, but we're not there yet.
And then a further question from Amit Pakai, who's talking about the issue of temporal updating, and again, to some extent, looking at the cohort in the earlier 3 months and then the last 3 months, and whether the prediction rates remain robust and stable. So I think you addressed that a little bit earlier, but and of course that could also be influenced by mutation and change in the actual propensity of the virus to cause severe disease. Any comments?
Yeah. Yes. So again, a really important question. We have had an initial look within our first wave data to see if there's any obvious signal of changes over time. And what we found, as you might predict, is that the discrimination through C was very consistent, but there was some suggestion of declining baseline risk over time because, of improvements in clinical management, perhaps because of some of our interventions that we knew were effective, such as dexamethasone. And so there was some evidence of declining baseline risk, between February and August. But we felt that the best way to assess that more definitively really would be a prospective assessment, which is why we're now assessing it in the second wave data with the preliminary analysis that I showed earlier. We'll be keeping a close eye on that over time, but so far at least we're not actually seeing any any dramatic evidence of a change in baseline risk, but certainly keen to keep an eye on that as things evolve.
Okay, and then another question coming in this time from Rob Hademan from Infectious Disease. Given that there may be further waves, let's hope not, is there scope to test the tool in an interventional clinical trial? And I guess that could be tested in a whole variety of ways. But
yeah, I think that's a really good question again. And to be honest, I don't really know the answer there. I think that sort of speaks to a broader question and theme, which is how do we really demonstrate whether or not these tools are actually useful and have impact in clinical practice? And I think that's a really important question to address. And I'm still not entirely sure what the best way to answer that question is. I think doing a randomized clinical trial looking for clinical impact overall would be really difficult in this field. With the example of 4C mortality, that has already been rolled out and a lot of places are using it. So I'm not sure what the feasibility of actually doing an interventional trial for that sort of thing would be prospectively, which is why we're looking at other sort of creative solutions to look at potential clinical impacts, such as analyses of RCT data to see if these might be useful ways to stratify treatment.
Okay, so I mean, if I can just follow that up with one of my questions, and please chip in, guys, if there's anything else. But so in relation to trials, obviously we participated in the recovery trial, and you know, in recovery, for example, with the dexamethasone, an early finding was that if you needed oxygen, then you got this 20% risk reduction, and if you went to ITU, you got a higher % risk reduction. But if you weren't on oxygen, and then you got steroids, actually, if anything, there was almost a signal of harm. And that's why getting that stratification right is important. You could, with your score, potentially go back to the patients who were recruited into recovery and actually have a look at that cohort of people who were treated with, for example, dexamethasone, and see whether you can tease out, even with a greater degree of granularity, who is genuinely benefiting from the dexamethasone based on their risk of deterioration. The point I'm making is that, you know, if somebody comes in and they're on oxygen, by definition, they get dexamethasone. Dexamethasone is not without risk, and yet that we don't know whether that particular individual for the next 10 days is going to get any benefit from it. We just base it on the average effect on the whole group. But if you could actually use a score and say, this individual who you're thinking about giving dexamethasone to, our risk prediction is they're not going to die anyway, and they're not going to deteriorate. Whereas this individual who's got a similar, you know, requirement for oxygen, actually they are at high risk of death and they are at high risk of clinical deterioration. So in many ways, that person getting dex is definitely going to get, probably almost certainly going to get some benefit, whereas the other patient may get something minor, but as they're not, nothing's going to happen to them anyway. I mean, that's, people don't realize this in the general public, but of course, you know, treatment effects are average effects in the average patient. But, you know, for every patient who benefits, there'll be a number of patients who get no benefit from that intervention, or indeed, in some cases, some harm. And I just wonder, I mean, whether, you know, going back and having that data set available and being able to predict their risk prior to going into the trial, whether you can see whether you can get even greater granularity. And that might be more relevant when we start looking at even more toxic drugs like tocilizumab and various other things, and whether we can really, you know, nail down the group that should really get these drugs. I mean, maybe a bit speculative. Others may want to come in. I think they do, but maybe you have your chance first.
Yeah, yeah, thanks, Brian. I completely agree with what you've said there, and that's why I'm particularly interested in that idea of looking in a post hoc way at RCT data to see if these scores might be more granular ways to stratify treatment, as opposed to, you know, the cruder measure of whether somebody's receiving oxygen or not, for example. I think it would be a really interesting thing to look at. Both, you know, you could either do it with some a priori thresholds using the risk scores, or you could even look at using the risk scores as continuous measures to look at whether there's obvious evidence of effect modification for the treatments of interest. It would be really interesting to look at that for steroids, remdesivir and anti-IL-6. And probably the best way to do that would be to, in a sort of individual participant data meta-analysis approach. If we could use pooled data across trials, that'd be really exciting. We're sort of putting in some initial feelers out in terms of feasibility of that. One of the big problems with recovery is that because it's such a pragmatic study, it doesn't capture quite a lot of the variables we need for the two scores. So there's been some talk about whether you could use data linkage or other ways to get around that. But so the sort of short answer there is we're looking into it, and I hope we would have some news over time.
Yeah, so David, and then Maddy, and then Chris.
That's brilliant. Just there's another factor here at work other than just the straightforward pathology, isn't there? You admitted roughly 15,000 patients to some form of ICU respiratory support, but 16,000 patients died, and the mortality on the ICU is not quite 100%. So there's clearly a lot of people who are dying well before they get escalated, i.e. we're palliating them or we're putting treatment escalation plans in place. And you do sort of mention that, but it's quite a large jump, isn't there, between death and those being escalated. And I appreciate that's going to be, age is probably weighing very heavily on that. But I do worry that the TEP and the palliation side of things has not necessarily been captured and brought through in your risk prediction.
Yeah, very good point.
Yeah, I think it's a really important point. And to be honest, that to us, though, was one of the important rationales of having the deterioration model alongside the mortality one, because, you know, we know that age is going to have a significant impact, along with other things, on treatment escalation plans. And therefore, we have this issue of the mortality risk often being sort of disproportionately low for some of the younger patients who are at high risk of deterioration. So I don't think there's necessarily a perfect endpoint here that encompasses, you know, that addresses that problem perfectly. And therefore, I think the best approach is to take into account the deterioration and the mortality risk side by side.
Yeah, and Chris, or Maddy rather than Chris.
Maddy.
Thanks. Just a very quick question around, or discussion point around clinical utility. In a moment, Dave and Chris, I'm going to ask you if you'd comment on how you might, in your respective roles as an ITU physician and an ED for AMU physician, how these scores might weigh in on your day-to-day practical decisions. But before I do that, just to go back to the opportunity of looking to see how we can use these scores to stratify outcomes with different interventions. The challenge has been to capture individual variables accurately. But actually, if we embed these scores in our day-to-day practice so that we're simply capturing the scores, the probability scores themselves accurately in all our patients going forward, that might be a relatively straightforward exercise in seeing at which sort of strata of probability, risk probability, different people might benefit differentially. So we could focus on the easier task of capturing risk scores rather than each of the individual variables, perhaps, going forward. So can I just sort of be cheeky and ask David and Chris how they might, in their different roles, take account of these scores?
Chris.
Yeah, I think that's a very fair point, and links in for me a little bit in what Jerry Brown was commenting on, whether one can sort of move that on and check it after 2 days and so on and so forth. So I suppose on the validation side of doing this sort of thing, one is presumably more tempted to use a score if the patient develops in the wrong way, and there may be an element of bias in the predictability here. I think for me, when I read all the scores, and I have to admit it's an age thing, and I'm sure it's the same for Bryan, that we grew up in medicine before an awful lot of scoring was available. And so we were much more used to look endlessly at patients, you know, hour after hour, and all the time scale during their admission to their inpatient stay to their discharge. But I think that these scorings are also very important from a management point of view, i.e. to just know how to deal with resources. How many patients are likely to need to go to an ITU? How many patients are likely to just need an IV? How many patients are likely to go to a mortuary and having enough fridge capacity? These sort of terrible but very important points. And I think maybe initially the importance of the score is twofold, i.e. A, 80% may be helping us to plan how we best utilize hospitals, and B, making us aware to maybe focus clinically our mind on the parameters which have a particularly steep curve, such as oxygen, such as this, that, and the other. The last point I like to make, maybe with the new wave, it would be also interesting to put a simple measure such BMI into the equation, because I certainly had.
You've gone muted, Chris.
A larger or thinner.
We missed that last pearl. What was that?
I can't hear you, Bryan.
You just muted for a while, but I think we got most of it. So, can I move on to another question now, which I guess we've kind of discussed already. It was where the patients who were recruited not part of any other trials, and I think the answer is many of them are part of other trials. And in many ways, that presents us with an opportunity. Is there any evidence, Rishi, that if you were part of a trial, that A, your prognosis was better in general terms, and that might be a selection bias, or B, that the quality of data was better? So in other words, there was something about being in a trial which meant that there were more parameters recorded on ISARIC, or maybe the centers doing trials were more likely to record more parameters on ISARIC.
To be honest, I think it's an important question, but I don't actually know the answer. And the reason is that within ISARIC 4C, I don't think there is an obvious indicator in the case record form as to which other trials they're in. So it requires some data linkage. They have been working on that, particularly to try and link the ISARIC participants with the recovery participants, so that we would have much more, granular physiological data for the recovery participants. But I think that's still a work in progress in terms of the last that I heard anyway. And
there's a couple of questions left, but we're almost out of time. But quickly on this one, and I think this is a good point, again, from Amit Bakai, data on NT-proBNP, although this is generally a cardiac biomarker for left ventricular strain, actually, I've seen some data on this, which looks a very strong predictor. And we're running the capacity program from UK for the UK from UCL, UCLH, and that is collecting enhanced data on people with cardiac underlying complications. And the most powerful cardiac predictor of risk is indeed NT-proBNP and heart failure. And I just wonder if was there not enough data in there? And also, Amit points out right ventricular as well as left ventricular strain, which may be relevant to pulmonary emboli. Have we got any data on that?
Yeah, important question again. So we basically, we don't have enough data completion for BNP within the ISARIC data, as you can imagine, on a sort of population level. It's not really being measured and captured in a standardized way across the NHS. So we just don't have data within ISARIC. But I think other data sources would be better, for example, capacity gen.
Well, Amit's offered to share the data with you if you want to try and plug some stuff in. And then finally from me, because of the justifiable issues around at least in the first wave, the realization that there appeared to be increased risk of deterioration and mortality in the BAME communities, Black, Asian, minority ethnic groups. Did this in any way come out in your score?
Yeah, so we did include ethnicity as a candidate predictor, but actually it was removed in our backward elimination exercise, meaning that it wasn't adding additional prognostic value over and above the other predictors that we've included. So that's to say that once we've adjusted for, their respiratory rate and their oxygen saturation, etc., it didn't add incremental value. But that's not to say that the BAME groups aren't at risk of more severe disease, but it's just to say that that did not come out as an independent predictor in our
Quite interesting. I mean, the big study from Leicester is showing the same thing. It was not related to the ethnicity, if you like, independently. It was more related to social circumstances and deprivation and various other things that contributed to increased exposure risk and therefore increased risk of developing disease and severe disease, which in itself is still an incredibly important issue. Okay, well, look, I mean, it's been great. This has been our first one. Thank you, Rishi. Thank you to the panel. I hope the audience have enjoyed it. I certainly have. And can I just remind you that we're going to try and run these now every 2 weeks for an hour at the same time, 12:30 to 1:30. The next one will be on the 3rd of February. And at that one, we'll have two presenters. One, Nikki Longley, talking about the PROVENT trial, which is this use of monoclonal passive immunization for patients. And the other one is Hanif Esmail, who'll be telling us all about the recovery trial. So they'll be both very interesting talks, and we'll adopt a similar format with a panel, and I hope you'll be able to join us. And finally, I'd just like to thank Farhat Galani and Matt Chorley from our BRC team and all those alongside them who've helped do the work in the background to make this possible, put this together, and ensuring that it's all worked and we've managed to get through it without any major hiccups. Thank you very much.